for those of you that are new to this activity, what I wanted to do was just let you know this is an open forum. In other words, students from across campus, you don't have to be engineers, you don't have to be biz econ majors. Anyone that is interested in joining this new venture competition is encouraged. And once you start, you'll see from this presentation, there are a number of support folks that are going to help you through this process. And we've got a lot of people we want to thank, but what we're trying to do here is take a student idea, and it can come from faculty research, and develop that idea with the help of lots of great mentors and lots of great faculty if you choose to take classes. And actually today you will see develop it to the point where it actually could launch as a company. It's pretty exciting. So what I'm going to do is just walk you through the process that these students have gone through in order to stand at this stage today, and we'll find out who are the winners of the competition. But I think in reality, any student that has participated, even if they're not up here on the stage, have really become a leader or are on their way to becoming a leader through the activities that you'll see in this presentation. So first what we do is we invite in any way we can students from across campus. You can be an undergrad, you can be a grad. We have a session where we invite the campus. We had about 130 students. And what we did this year, which was unique, is we took mentors and judges from past years and had them stand up and talk about what they felt was the opportunity and what it was like to be a judge. And so these are the people that volunteered their time on that night. And then we also had Susan Block. You'll see Susan Block volunteers for a lot of these evening events, talk about what you need, what are the elements of a presentation, just so the students were aware, okay, first thing I have to do is stand up in a room of 200 people and give a presentation. Then what we have is every week thereafter for about 7 weeks, every so often we skip a week, but you have 7 different weeks where we have a mentor night. And what that means is the student teams, after they assemble, go to this large room, and there's stations with mentors with varying backgrounds. You will get to see we've got an exceptional group of mentors. And in 20-minute rotations, the students talk about their idea, and first, you're gaining a network, and second, you're gaining a lot of expert opinion. So the first night we had was our marketing night. The second week later, we had a finance night. The third, we had a technology mentor night. So one common theme and a consistent theme with this program is the competition isn't just about a general idea, it has a lot to do with technology. So you can be a student from across campus, anywhere, and meet these mentors and go through this whole process, but you're not going to be standing up on this stage if your idea was something not technology-based. For example, a better way to market, a fast food chain or a new fast food chain. Okay, at that point, they've had a few mentor evenings. They've also gotten a presentation on how to give a pitch. We assemble a fair, and what this is the students have tables like they do right outside of this room. I hope you visit those tables during our reception. And the students give pretty professional pitches. And the people that are attending the fair, we had about 30 people at this fair, and it's past winners from the competition, and investors and service providers. And by the end of the evening, we started with 9 teams. We have certain investors put their TMP bucks in a box, and we count them, and whatever teams received the most votes made it to the finals today. Once we knew who was going to proceed on, what teams were going to proceed on, we give each team, now keep in mind they've had all these mentors, hopefully they've gained a lot of business cards and they're using these other mentors, But at the end of our semifinal venture fair, the teams then get assigned, based on their input, a longer-term mentor. So this longer-term mentor, or sometimes two mentors, depending on the composition of the team, really is going to help their team succeed. And by doing that, you may actually become the CEO of, or an interim CEO of one of these companies. There's a lot of past mentors who have proceeded on to help these students actually start companies. So this is just the names of of each of the team's longer-term mentors. Oops. Okay, so then after being assigned your longer-term mentor, they go through an additional process where they get more information on giving pitches. Then they have to, over about a month period, refine their presentation in front of a large panel. So they actually met with two separate panels, one on May 3rd and one on May 19th, to give you kind of the end product you'll see today. And these panels are comprised of people that do this professionally. So what I'm hoping you see is that this program is really a program built from you and the community. It's about how you can help students become leaders and maybe even found companies. So what I'd like to do, because we've got probably half the room are people that have been involved in the TMP program from one spectrum or another, can you please stand up? Don't be shy. I see lots of mentors out there. Don't just sit there. Oh, come on. And I'm not just talking about this year. I mean, there's lots of people in the room that have volunteered over the many years that we've been doing this. And then, is David Kremen in the room? Okay, well, what I'd like to also announce is at the end of this, there are a number of follow-on competitions that are certainly chasing after our winning teams. And the very next competition is one, this is their second annual event. A venture firm, Draper Fisher Jurvetson, actually takes the cream of the crop from the colleges in the state of California. And they have another competition, so if you're really interested in these things, you're welcome to go. It's at, up in Mountain View. And then they also have an opportunity to join Fortune's small business competition.
Good afternoon. My name is Sejal Hall, and along with Marco Mena and Kevin Bulwer, we are Aware Biotechnologies, developing the next generation of cancer diagnostics. I'm going to talk to you today about our business opportunity, and Marco's going to discuss our business model. Breast cancer is one of the most prevalent forms of cancer. In fact, 1 in 8 women will be diagnosed with it. However, as is known for many other cancers, early detection leads to better outcomes. For example, if you can diagnose breast cancer in stage 0 of the disease, the survival rate is 95%. Compare that to a diagnosis in stage 4, where the survival rate is less than 10%. Because breast cancer is so prevalent, mammograms are recommended as an annual screen for women over 40. However, mammograms are expensive, costing anywhere from $100 to $200. They're very uncomfortable, and they're inconvenient. Women may wait up to 6 months to get a mammogram because there's a declining number of certified mammography clinics in the US. All this leads to low patient compliance. Women who need to be screened don't get screened. Therefore, breast cancer is diagnosed at later stages, and we have poorer patient outcomes. Our solution is a blood-based breast cancer diagnostic test, similar to the PSA test for prostate cancer. We will sell a detection solution that, when mixed with patient blood, will give off a fluorescent signal only if the patient has breast cancer. Our test is inexpensive. It's expected to be offered to patients for anywhere from $25 to $50. It's non-invasive, requiring only a patient blood sample, and it's very easy to administer. Now, we believe that all this can lead to increased patient compliance. Therefore, women will be screened regularly. We can diagnose breast cancer at early stages and have better patient outcomes. The test is developed from our core technology, which was developed here at UCSB by our CTO, Kevin Boulware. It's a biomarker discovery platform that enables us to find molecules that can distinguish disease samples from normal samples. The molecules can then be packaged and sold as a detection solution. Now, these molecules are actually detecting levels of disease-specific proteins, so not only can they be used for a diagnostic test, but we can also track a patient's response to therapy and also track disease progression. The technology is not just applicable to breast cancer, but it can be used for other types of cancers and other diseases as well. So although breast cancer diagnostic is our first target product, we will develop products for other types of cancers and other diseases. And you can also imagine that these tests may be performed individually or together in a multiplex format. The technology is patent pending, and a proof of concept has been completed. The results of that study were published earlier this year in the Proceedings of the National Academy of Sciences. The market opportunity for breast cancer is 70 million women in the US alone. Now, 42 million women of those are currently screened annually, amounting to over $4 billion spent on mammograms in the US. Now, while that's a significant market already, there's still a large fraction of women that aren't being tested, and so we feel that there's opportunity for growth as well. In addition, 40% of all people are diagnosed with cancer. Therefore, we would like to expand into other cancer diagnostics markets, especially for cancers like ovarian and pancreatic cancer. They have very high mortality rates, and there are no existing screens. Therefore, we feel we can actually build markets in those areas. The competitive landscape for breast cancer diagnostics currently consists of non-biomarker substitute products such as mammograms and the self-breast exam. However, these products suffer from problems with cost, invasiveness, and accuracy. There are also smaller firms that have directly competing products. All of these companies are developing biomarker-based breast cancer diagnostics. However, none of them have an FDA-approved test. In addition, none of these companies have a biomarker discovery platform. Therefore, their tests are based on biomarkers that are known to be associated with all types of cancers. They're not specific to breast cancer. Along those lines, the companies also have problems with the cost of their test. The tests are also difficult to administer, and they're not scalable. Therefore, we feel we have significant advantages over these products. Finally, there are large companies in the diagnostics market, such as Roche and BD. These companies are focused mostly on developing instrumentation to perform the diagnostic tests. Therefore, we actually view them as potential partners rather than competitors, since our tests can be easily integrated into their existing instrumentation.
Our product development timeline is as follows. The first year will consist of research and development and early stage validation of our markers, at which point we can actually sell them labeled for investigational use. In order to sell our markers as diagnostics, we will go through an FDA pre-market approval process, which we expect to take 12 to 15 months. Once we obtain an FDA approval, sales and marketing of the product can be done not only to the labs that we sell to directly, but also to the doctors who are responsible for ordering the test and to the HMOs who are responsible for paying for them. Now, the nice thing about this model is that it can be applied to different tests. Initially, we plan to focus on breast cancer, but then the very same model can be applied to develop ovarian and pancreatic cancers diagnostics which have unmet diagnostic needs. Now, as these three products come online, we expect to generate over $100 million in revenue by year 10 and approximately $30 million in earnings. In order to get there, we are going to require $3 million in 2 rounds of financing. The first is a small round of $500,000, which will be composed of a mix of grants and equity and used to finance product research and development, as well as validation and at the small scale. Subsequent to that, a $2.5 million round will be used to obtain clinical pre-market approval from the FDA. And then we expect to be cash flow positive by 2009, at which point we will use revenue to generate future growth. Our management team currently consists of the three of us, who are all PhD candidates here at UCSB, as well as two professors from UCSB, one of whom has experience with a startup that has since gone public, as well as an intellectual property specialist and a medical doctor. In summary, we have a technology that we feel fills a clear market need, allowing for early detection which improves outcomes, saving patient lives and money. We have a patentable platform technology that greatly reduces the discovery time and allows us to generate additional intellectual property from each candidate marker. The technology is applicable to multiple cancers and other diseases, either in standalone or as an integrated multiplex product, which allows us to have multiple revenue streams and reduce the risk of any one particular indication. Finally, not only is a product important in that it saves lives, but it gives us a favorable financial outlook with early and scalable revenue and cash flow. And with that, I'd like to address any questions that you may have. Thank you.
There are a number of different types of breast cancer, and it's difficult to say that there's likely to be one biomarker. And I think you could say that's probably true for many different kinds of cancer. So it would seem to me that you might end up having to do something where you end up with a panel of biomarkers and the relative response to them create a pattern that suggests cancer, which then you do follow-up tests, et cetera, which start other costs going. That just seems like, if that's true, it just seems like a much bigger project than you can possibly do in that short a time frame and with that little of money. And so I just wonder, what happens if that turns out to be true?
So the nice thing about our discovery platform is actually that the way it works is you pool samples of patients with breast cancer of all different types, and then patients that do not have cancer. And you seek out the molecules that are in that sample. Now, you don't, in an ideal world, you would find one molecule, but the platform also allows you to get a whole panel of them, in which case, and then you can use the same technology to go back and validate it with individual patient samples. So should it be a panel instead of a single molecule, our technology has the capability to capture that.
So you'd separate those different molecules so you could read each one, because otherwise you'd have a real problem with signal to noise.
Correct. And it would basically mean that you use 2 wells instead of 1, or 8 wells instead of 1, and that isn't a large difference. Or we don't expect it to be.
How strong and unique is the IP? Apparently you're using this CLIPS technology, but it's part of the UC system, so couldn't others use the same IP?
We're in the process of hopefully licensing it exclusively from the UC. Whether our business model kind of would be improved a lot more if it is an exclusive license, but we're currently in the process of working out those negotiations. And we can generate, once we have a license to even use the platform technology, each subsequent biomarker would be owned by us.
I'm having a hard time understanding your timeline based upon looking at the competitive firms in the marketplace. If you look at IBEX, Power 3, or biomarkers, all who have been in the marketplace and funded for a while, you know, one of them being publicly held, and yet you say that none of them have FDA approval. So what is it about your technology that makes you believe that you can get there that fast?
So the simple answer is the platform and the validation would go hand in hand, rather than these companies have been around for a while, spent their first few years trying to find biomarkers, then the next few years validating the biomarkers, applying for FDA, and are just in the process of starting the clinical trials and going through all of those things. I'm sorry.
So you're saying that all of these companies started without having any target drugs or concept drugs or no biomarkers, no nothing.
Right.
What got them funded? I'm being serious.
Yeah.
I mean, in other words, what did they, on what basis did they get started that, that would have gotten them? Is there a good reason?
So IBEX, for example, isn't a biomarker technology company. They're just recently trying to develop ovarian
Okay.
cancer biomarkers. They've already sold a big part of their technology, I think, 10 years ago.
Okay, so then we'll go back to your answer. So you're saying you already have the biomarkers in hand, so it's a validation and platform development.
Again, Kevin?
No, we have the platform.
You have the platform?
We're going to develop, we've had the proof of concept for model proteases, and we will develop breast cancer specific biomarkers. That's what our plan is.
Okay.
And it's a different method, I guess, than how they're finding biomarkers. They use a lot longer process. Takes many more years.
Okay. Is the cost you're projecting for the test similar to the other tests like the PSA test and things like that?
That's what we're targeting.
Okay.
So what do you think the requirement for accuracy is in order for this to be approved, and what makes you confident that given the diversity of the disease that you can actually achieve a sufficient level of accuracy to make this a useful test?
So the interesting thing about your question is the concept of accuracy, which is both, it's kind of a two-dimensional sensitivity and specificity type of question, where you don't want false positives or false negatives. The thing about this technology, because you're measuring levels of a particular molecule, you actually develop a response curve that is a trade-off between sensitivity and specificity. And the FDA requirements as to what the minimum is would depend on what is the established technology, which is mammograms, which compared to other to known blood-based biomarkers actually have a pretty low sensitivity and specificity. So the bar is set somewhat low because the technology that is currently available is in many cases inadequate. Numbers are a little difficult to get to without talking about the trade-off, but it's if you can detect over half of, like a false positive rate over 50% or something would be something that the FDA would consider.
But I mean, you need to have a specific target so you know whether you have a product or not, right?
Right, and
So I
that's
guess
part
what
of this
I'm asking
first
is
validation.
What do you need to achieve? What is your target, and how confident are you of and why are you confident of your ability to hit it?
Right, I think that those questions are addressed in the concept of this next small-scale, bench-scale validation, where we set a specific get the response curve, and figure out what sensitivity and specificity values are required.
Assuming it doesn't happen quite so quickly, and so your accuracy is not there yet, have you modeled the business as a supplier of research reagents, of things for people to look at and study further for a few years and see if you can keep going that way until you get to the right combination?
Right, so we haven't done detailed modeling of that, but that's definitely in our plan. As I said, prior to receiving FDA approval, you can sell the reagents for investigational use, and we expect to do that as soon as we have confidence that the reagents are sellable. And there's definitely cases where companies basically maintain themselves and their cash flow by going to this kind of investigational model, and that's something that we would probably do if during the course of obtaining our approvals.
Well, thank you.
Cliption is a biotechnology company positioned at the interface of molecular biology and material science, positioned to deliver the next generation of drug delivery technology. Current protein-based therapeutics are able to attack targets on the outside of cells. And while these have great utility, they're more available and more useful targets that exist on the inside of cells. Nucleic acid-based therapies alone are able to target these, and are able to provide effective therapeutics for cancer, diabetes, autoimmune diseases, adult onset blindness, and viral infections. There's a problem though. While nucleic acid therapies have tremendous potential, they won't work from the outside and require delivery to the inside of the cell. What's the solution? A delivery vehicle. Cliption Biotechnology sells a delivery vehicle that is able to self-assemble around the nucleic acid payload, much like a self-assembling smart missile. and deliver the nucleic acid payload to the inside of cells, thus removing disease-causing agents. This has implications for both research reagents, for drug discovery, and as a therapeutic. Current market needs are that small nucleic acid therapies are particularly useful as both basic research reagent and for drug discovery, but they're also particularly difficult to deliver. Current formulations for delivery are inefficient, toxic, and unstable. Furthermore, in terms of basic research and drug discovery, only a limited number of cell types are able to be accessed by current delivery reagents. The current strategies that people use for delivery of nucleic acids to cells are centered on one of two methods. The lipid-based reagents, which are structurally very similar to the outside membrane of a cell, and have the benefit of high fusion with the cell, much like 2 drops of oil coalescing in water. The dendrimer-based technologies have the benefit of a very high positive charge. It's able to bind the negatively charged DNA very strongly and provide high stability. Calypso's unique technology fuses the 2 types of molecules into one, giving the benefits of both and the detriments of neither. This has the advantage of being more stable, less toxic, and more efficient. This has benefits for R&D in terms of basic research and drug discovery, and a lower cost and time of drug development, and for the delivery of therapeutic-based nucleic acids in longer-lasting, less toxic, and more efficient drugs. As the Calypso delivery reagent is a platform technology, it has the ability to address 2 separate markets. The first market it has an application in is for the delivery of nucleic acids as a research reagent for basic research and drug discovery. This is a market for drug delivery that's currently estimated at $160 million per year and growing at 30% per year, reflective in the tremendous enthusiasm for the potential of small nucleic acids for drug discovery. It also has an application as a therapeutic, and the addressable market for that is projected to be $600 million. There are currently 14 companies that have been assembled with a mission of delivering the next generation of nucleic acid therapeutics based on small RNAs. It's projected if even a few of those therapies come to market within 2010, the estimated value is going to be $600 million. Furthermore, each one of these companies has multiple drugs in their pipelines. The advantage of commercialization of our delivery reagent for basic research and drug discovery is that it's a market that's much easier to access as there's no clinical trials required. Furthermore, our first product is ready for market and has already received demand. Finally, as we have an adaptable core technology, our second generation of research reagents is already in the works. All it's needed is scale up manufacturing of our first product and obtaining a channel partner for distribution. The customers would be biotech R&D labs, as well as basic research and academic labs. This has the advantage of rapid revenue generation, with revenue anticipated in year 2 and up to $30 million potential revenue by year 5. Application of our delivery technology for the delivery of therapeutic nucleic acids has a benefit of a much larger potential reward when the products come to market. However, it's associated with a much longer development cycle as clinical trials are a necessity. We do expect early revenue to come in the form of upfront payments and milestone payments, and later, but much larger revenue potential to come from royalty payments on sales of the nucleic acid-based drugs. We anticipate as much as $20 million in revenue in the form of upfront milestone payments by year 5 and $250 million by year 12. Based on the combination of marketing of our drug delivery vehicle for the purpose of basic research and as a potential delivery of therapeutics, we anticipate revenue in year 2, becoming cash flow positive by year 3, breaking even by year 4, followed by rapid growth from year 5 onwards. We're seeking $4 million in total funding, which will go towards acquisition of an experienced management team, including an experienced CEO and Director of Business Development, the scale of manufacturing of our first product for the research reagent market, as well as further R&D into development of second-generation research reagents, as well as for the delivery of therapeutic nucleic acids. Our core team is composed of 4 graduate students with differing backgrounds in chemistry, molecular biology, and material science that together provide the synergy necessary to drive the innovation for the next generation of drug delivery technology. Our board members, scientific advisory, and board of directors are composed of the primary researchers and principal investigators responsible for generation of this technology, as well as individuals who have vast experience and vast and diverse expertise in the commercialization of biotechnology products. In summary, we offer a platform technology that has application in 2 separate markets, each with differing risk-reward profiles. We anticipate early revenue based on sales of our research reagent, and later, but much larger revenue from collaborations with drug companies looking for a delivery agent for their nucleic acid drugs. Our first product is already ready for market. It's proven to work, and there's already demand for it. Furthermore, we have our next generation of products in the pipeline. We thank you for your time, and we'll field any questions.
So what are the competitive technologies for the delivery in the therapeutic market?
Technology expert.
Currently, for the delivery of small interfering RNAs, which are the small nucleic acid-based therapies, what Rafi showed on one of those slides is there's really 2 fields of thought. There are lipid-based technologies and basically synthetic polymer-based technologies. So there are competitors who make both of those products. And I guess we're a fusion between those 2 products, and we've created a whole new molecule based on those 2 technologies together.
So how big is the market for those two competitive products today?
It's $160 million per year for research reagents.
But for therapeutics?
For therapeutics, there are currently no therapeutics on the market, as this is a brand new technology. It's much like the monoclonal antibody field was 10, 15 years ago.
So
probably more than that.
My experience, limited though it may be, with new treatment modalities like this is they take a long time to get through the FDA process.
There are already drugs in phase 2 trials, actually.
Already?
There's 14 companies that have been organized to capitalize on this technology. This is sort of the latest generation.
Being small
nucleic acids,
small nucleic
not
acids.
delivery.
but through different mechanisms.
Right.
Yeah,
But none of them are in the market today. They're in phase 2.
that's correct.
So what's your forecast for the realistic time frame for your product to get through the FDA trials? And the probability of success, since, the fallout's high.
There's already successful animal data with the application of this type of technology for the delivery of small nucleic acids. Our core technology hasn't shown any overt toxicity in a different sort of formulation previously. Can you guys think of any?
In terms of success, it's interesting to think about how you would define success in this field, because the way the technology works for these 14 companies is they really are trying to find targets. So they have this small piece of siRNA, which has a certain sequence. It's the sequence which is important to them. So the sequence determines whether or not they can cure a certain disease. So they can make tons and tons of different sequences, but our product will actually work for all of those sequences. So as long as one of their targets is successful, our delivery reagent has a much greater chance of success.
Can you give an example disease where there's a specific nucleic acid target thing that you're trying to deliver and what it would do? Or are we basically talking about a whole class of diseases which, at least in theory, can be addressed this way?
This technology's application actually to any disease where there's a gene that's able to cause that disease. So for instance, for a viral infection, it's able to target viral genes. For cancer, it's able to target a class of genes which are currently only able to be targeted by making synthetic molecules, much like the Gleevix, I don't know if you've heard of those, and manipulating targets from the inside of the cell. As I stated in the presentation, there are far more available and useful targets that exist in the inside of the cell that determine cellular growth, and the ability to cause cancer. So this technology, nucleic acid technology, has potential to cure and provide effective treatment for just about any sort of disease you can think of, from cancer to viral diseases to autoimmune diseases.
See, going back to Bill's point about how long it takes to get a therapeutic through, the FDA approves specific treatments for specific diseases.
Yep.
So you can't just go in with a class this can do a whole bunch of things. You've got to come up with some specifics. So it seems to me we're talking quite a few years.
Well, they already have these small RNA molecules that target, for example, macular degeneration. and they target a specific protein that's not functioning properly. These drugs have been developed by a couple companies, and there's one that's already in phase 2 clinical trials right now. So basically, they are progressing along the timeline. They aren't just theoretical therapy.
The main advantage really is that the impetus isn't really on us to develop nucleic acid therapeutics. We're just really the delivery vehicle, and we can work with the nucleic acid therapeutics from any one of the 14 companies that have already been organized to deliver these.
Yeah, but if there's nothing to deliver, it doesn't matter.
Oh, no, that's not an issue. Yeah, these have been proven to work in cell culture, and even in the absence of delivery, what's usually necessary, for instance, for macular degeneration, is direct injection through the eyeball, and that already has been proven to work, as well as for targeting a particular type of viral disease called CMV.
Yeah, it's not so much the technology itself doesn't work. Like I was saying, these companies really need to find the proper sequences and the proper targets in order to carry out this technology of small interfering RNAs. So the technology itself does work as a therapeutic. The companies need to find the proper targets to see what proteins they need to knock out. And then we step in as to provide a delivery vehicle for the molecule where they find the target to actually work with success.
And all this searching is done in cell culture, where the reagent market that we're planning on developing has utility. So basically our molecules could be used in the research reagent market to actually test these potential therapies.
Target validation.
So again, in this first market that you don't need the FDA approval, I mean, what is, again, so unique about what you're doing versus the people that are out there today?
It's the fact that we've combined this dendrimer head group with the lipid tails. So for these nucleic acids are so small, it's very difficult to make a stable complex. So that these things want to fall apart just naturally due to the very small size of the nucleic acid. This is just due to some entropy. So because we have a dendrimer on the end of our lipid, we can create a large density of charges. And it's the result of this charge complexing with these negative nucleic acids, which create a highly stable complex. So nobody else out there in the market has the charge density that our molecule has. And it's essential for the small nucleic acids to have this high charge density so that you can create a stable, lasting product.
And this, because you need less of our molecules, basically you end up with less toxicity, because these our competitors' lipids, you need a substantial amount, and in the end what you end up with in experiments in cell culture is you end up with a number of cells killed that you wouldn't have if you could have something where you could get a stable complex with less of your lipid.
There's a quote actually I read from one of the people in upper management at Stratogene, who are a competitor that make the same, a similar sort of product, but without the advantageous properties. And he said that when you're doing these experiments, trying to get a nucleic acid into cells is really hit or miss because of the high toxicity. Either you get it in and you kill everything in the test tube, which means you have to start over again, or it works. So low toxicity and high stability are very advantageous properties to have.
All right. Very.
Thank you very much.
Thank you.
Good afternoon. My name is Aaron Levy. This is Nathan Level and Adam Brenner. Scott Perry couldn't make it here today. We are Emergency Medical Technologies, a medical advice company. There's a very serious problem right now with the current trauma procedure. Keep in mind, trauma is the #1 cause of death for patients under the age of 44. If a patient is unconscious and is not breathing, the life-saving procedure that will be employed is called endotracheal intubation. And it's a process in which a tube is inserted into the windpipe of a patient for ventilation. The way this is done is with a medical device called a laryngoscope. It allows the practitioner to visually see into the throat to see the vocal cords to insert the tube. If you cannot see the vocal cords, this procedure cannot continue. And oftentimes in trauma cases, about 30% of the time, blood or other bodily fluids are in the throat causing this visual hindrance. If this is the case, a suction device will be implemented to remove those fluids. For instance, if you're in the field, a VVAC will be used, which basically evacuates the throat. Due to its blind nature, however, it takes 15 seconds and it requires usually 2 people because your hand will be having endotracheal tube and then your laryngoscope in the other hand. We have solved this problem. We have combined these two ineffective devices into one efficient medical device we call the vacuscope. It saves time by having not to switch between devices, as well as it's more precise. It reduces cost by only having to buy one device, and most importantly, it'll save more lives. Our engineers have developed innovative technology to combine these two devices. Currently, a laryngoscope has two double-D batteries within the handle that power a light at the edge of the blade. By replacing that light with an LED that's brighter, we can use two small watch-sized batteries and have the entire handle for use. Within this handle, we employ our technology, which is a venturi device powered by a compressed gas canister and a trigger device. We have a finished prototype that is, however, non-disposable, but the final device will have a cartridge that is disposable, including the venturi device, tubing, and the cartridge. This will allow current sterilization procedures as autoclave to be performed, and furthermore, since ergonomics aren't really changing with our device, no further training is going to be necessary other than learning how to press a button. There's a very attractive niche within the medical community or market that's $590 million US yearly for laryngoscopes that are non-surgical and suction devices that are used for just endotracheal intubation. Furthermore, there's also a $2.9 billion market for people who have laryngoscope that don't need to be replaced yet due to wear and tear. But because our device adds so much essential and vital time to this procedure, would be also likely to switch their device sooner. Furthermore, this problem is not unique to the United States, and international markets are also a future potential market. The business model we plan on employing involves using sales through a national distributor. Aside from being the standard for such a device, these people already have the connections between all of the hospitals nationwide and the relationship with people who do the purchasing. And moreover, we're going to plan on doing outsourced manufacturing to keep costs low. Because the parts internally are very easy to manufacture, this should not be a problem, and there are many manufacturers to do this process. But to ensure that we have quality and control, we will be doing in-house assembly of our devices. The customers we plan on targeting include hospitals foremost, considering they hold 70% of the market, but also fire trucks, ambulances, other EMS providers, as well as the medical training facilities where people learn how to use this device. The competition we face include both the laryngoscope and the suction device manufacturers. Welch Allyn sells about 70% within the first category, and Lardell second, selling the VVACs and other portable suction devices. However, we will be able to compete within this market because our product is more efficient. It'll save precious time when a patient is not breathing. We will have intellectual property, hopefully keep them away, at least as a defensive tactic, and will be of lower cost to the purchaser. According to Ron Worf, the CEO of Cottage Hospital and also one of our advisors, as well as his chief purchaser at the hospital, there are two main ways a new medical device enters the facility, including internal pressure from the staff, as well as through advice from medical analysts. VHA is one of these analysts. They advise 1,600 hospitals nationwide. And because we're providing such a good technology that saves such precious time in a life-saving procedure, we believe we'll have high ratings. Furthermore, we plan on giving our device to select trauma centers that have a high ratio of EMS trauma volume, as well as to, since there's such few paramedic schools, to give them as well to them since it's cheap to manufacture so that they can train their paramedics. The management team includes myself. I am a chemical engineer and a business technology management emphasis. Nathan Lavelle and Scott Perry are graduated mechanical engineers. Adam Brenner, who is a business economics student. We've been very fortunate to have assembled an all-star advisory board that span the entire spectrum of bringing a medical device to market. This ranges from the CEO of Cottage Hospital to the director of clinical trials at UCSD to two successful founders of a medical device company. We've had great help building a financial forecast from one of our advisors who's a CFO of 3 successful companies. And with this, we believe that with 0.6% of the market or less than 10 hospitals by year 3, we'll be able to break even. Furthermore, if we have 8% of the hospital or less than 142 hospitals by year 5 within the market, we'll be grossing sales of $48.5 million and a $21.1 million in net profits. The required investment to reach these goals include first, a $500,000 seed investment to get the mass production final design for cheap mass production, attaining the intellectual property, beginning the process, hiring a CEO who's had experience bringing a product to market, as well as a director of quality control, who will begin our FDA 510K process. And at that point, we believe we'll be ready for the second round, which is $1.5 million, and it'll be the final round. With this, we plan on going to market. To recap, there's a strong need for our medical product. We have a completed prototype, there's a large potential market, and we have a solid product. Our product will lower costs for the purchasers, save precious time for a patient who's not breathing, and more importantly, it'll save a lot of lives. At this point, I'd like to thank you and take questions from the judges.
It seems like a terrific idea.
Thank you.
It's straightforward. The only question I guess I have is, how really unique can you keep yourself? Because these other guys know how to do suction. You're combining these two pieces, right? And you figured out a way to be a little bit more innovative. But I guess how unique and defensible are you? Because other people are going to try to copy you.
Very unique. Nathan, who's the mechanical engineer, will explain.
Actually, the way we've done it is we've used a Bernoulli device. Other people have tried to use mechanical systems and they've failed. Honestly, the closest thing is this thing. It's about the size of a computer monitor that they have to carry around. Those things really don't work, and that's why they're very expensive, and that's why they're not very popular. Our device, on the other hand, we will get a patent on specifically suction inside of a laryngoscope. That's going to block out everybody else from the market, and honestly, we feel that that's a strong enough patent in itself. In addition, we're going to be getting several design patents, and we also want to get a patent on the disposable cartridge. Regarding how unique our technology is, we're using a Venturi device. This allows us to generate suction off of compressed air. This means that we don't have batteries, we don't have power systems, we don't have mechanical systems. When we started designing this in the first place, my engineering team and I, we tried all sorts of different mechanical systems. We thought these mechanical systems would be the best route to go. Turns out they're not. They not only don't work, but they're very expensive to manufacture and they break very easily. We broke several prototypes real quick and we never got the prototypes to work in the first place. This device, on the other hand, is cheap for us to manufacture and it works very, very well.
What's the suction level on the device compared to the other suction devices that are in use? What kind of...
I'm glad you asked that. Currently, we're actually able to achieve suction rates of over 10 milliliters per second. Now, in the hospitals, they're getting of around 30 to 40, but the difference is that those are designed specifically for pumping stomachs. When you're using the ones that are for endotracheal intubation, the blind ones get ridiculously great suction levels, but they fail a lot. And the reason is, and we'll show you a demo outside if you come out later, it's a large tube that's about the size of your esophagus. Therefore, when you use it to suction, you can't see what you're doing. You're vacuuming out the person's stomach, probably their breakfast from yesterday. Unfortunately, all you really want to vacuum out is the area that covers the throat right here. We're suctioning out from specifically the Adam's apple up. That's where the trachea is. These other devices, you actually jam down into the throat around here before you can even start suctioning.
Okay, I've got more questions. Go ahead, Bob. Yeah, lots of questions.
Kathy's going to work you over pretty good.
When I actually understand.
A question about approach. It's clear that the design and the engineering, and particularly the feature set that you choose, is critically important, and yet it looks like from the business plan that you're outsourcing all of this, and particularly the engineering, which seems to me to be very risky, and it saves some money, but it could cause 2 or 3 design cycles when you could have got away with 1 or 2, that sort of thing, really, time to market, et cetera. So I just wonder what's the thinking about doing it that way rather than trying to do it in-house.
Well, we're about 90% done with the prototype. The prototype works, however, we don't know how to design to have a cheap mass manufacturing process. So basically what they're going to do is take our design that works and just make it so it's cheap to manufacture.
Who's they?
There's a few companies, I can't remember the first one. Inogen, do you remember the company in Irvine that you guys used? Omnica.
Okay.
Specifically on that, like he was saying, our engineering team, we're graduating engineers. We know how to build a device that works, but we don't really know how to make it something that's easy for mass production. This is why we want to contract a firm that's specialized in this, that's experienced in this, because sure, we could make our device mass produced. We think it would be a lot more effective and a lot cheaper for us to produce if we sent it to one of these firms first. We think we'd recoup the cost very quickly.
It isn't a question of cost. For example, one of the decisions, it sounds like, is that you're going to use a blade or a set of blades that are autoclaved, whereas the others all have blades that are disposable. I don't know, who decides what, that sort of thing, what's best.
And go talk about the blades.
Currently, there are several types of laryngoscopes. There are some disposable ones, like the ones that you're talking about, and the majority are autoclavable. The current problem with the disposable blades, there's a bunch of problems with them. They're one of the greatest ideas that doesn't work, kind of like the spork at Taco Bell, the combination between a spoon and a fork. It just doesn't work. What ends up happening, we actually talked with a firefighter down to UCSB, and he was complaining about how their blades tend to bend. They have to be really thick. They block the view. They're not going to use those anymore. I'm now a paramedic student, and I was talking to a couple of my paramedic professors. They were talking about the blades that they have that go through temperature extremes and end up warping real quickly, so they can't keep them for very long. They have to keep a very small number. Disposable blades jumped into the market for a short time, and they're getting phased out real quick. Everyone's getting rid of them. Specifically, the disposable part on our device is the stuff that would be impossible to clean, such as the Venturi device. That would be very expensive to clean, especially out in the field, but also in hospitals, because it's not an autoclavable system. Since we're already going to have to get rid of the tubing and the cartridge, we can build these Venturi devices for dirt cheap. These are going to be made out of plastics, and they can be molded. A few pennies for those, and that way we can make money on cartridges and also save the hospitals the money on the cleaning systems.
You say you're going to be able to make this very cheaply, and you have a really high gross profit in here. Did you notice that one? And so, but it sounds like you haven't finished the design. So is this, I mean, what makes you think that a GP of 90% is realistic?
The cost on the design right now, we were given a budget of $2,000 to build this product from start to finish. This is including all the failed stuff that's gone on with this device. It hasn't cost us $1,000 yet, and that's with all the research and development. This current device that I've got outside there costs us about $20 to manufacture, and that's with us using metal parts. That's with us using store-bought parts that cost $12 in and of themselves. We also weren't quite sure about those costs, so we went and talked with
Craig Spear, the Director of Engineering at Conman Limitech.
Familiar with him?
He actually told, he told us that the numbers that we had were actually probably too high. He said that we could probably produce this for even cheaper.
Okay, so you priced it competitively with what is existing in the market today. How have you priced it compared to existing laryngoscopes? I wasn't really clear on that.
Well Challenge sells a 4-to-1 ratio with the other companies that make laryngoscopes. Hind is the other one, and then the last one of the 4-to-1 is every other company. They average price is $582 for the laryngoscope.
Okay, so that's fine.
And the suction device, the portable mechanical one, is about $675.
Okay. I'm sure it was in there. I'm just trying to remember, go back on these new financials, so, okay.
In addition to that, when we were coming up with the cost, we at first had actually a slightly lower cost, and when we talked with Ron Worf, the CEO of Cottage Hospital, he said our cost was too low, that we could actually jump the price up above the $800 that we have it at right now. He was encouraging us to charge more.
Wait a minute. I know what it costs to run the hospital. I don't think it's a good idea. How about sales channel?
Conflict of interest.
So what's the regulatory process that you're required to go through for this, and what makes you think you can do it in the time frame you have allocated?
We spoke to the regulatory guy at Conman Limitech. The laryngoscope is a class 1 510K. That means that you don't need to do a 510K. It's exempt, but you still have to have the regulatory control for your like assembly, for instance. There's a possibility it might be a class 2, is why we gave it a year and a half to process that. And one of our second hire, aside from being a CEO, we need a person who's a director of quality control and has had experience and can save us the pitfalls of having to go back and forth through the FDA.
Thanks.
Thank you very much.